Alright, hi class. My name is Sarah Wilkins. This is Advanced Group Concepts presentation number 3. Today we're going to talk a little bit about oxygen binding to heme. Hemoglobin is a protein which binds to oxygen in the lungs and releases it in the capillaries. Our body controls this intake and drop off of oxygen by having different pHs at 2 separate locations. So for the goal today, we're going to talk about how oxygen binds to the heme groups and structural changes that occur, along with the binding capacity of oxygen to heme and what differs from that in the capillaries. Also going to talk a little bit about the CO2 interacting with water and how that essentially can affect the pH of the capillaries. Alright, for the heme groups. So hemoglobin has essentially 4 different heme groups that are bound to the globin. There are 4 square planar coordination positions of Fe that are occupied by 4 nitrogens of the protoporphyrin. The heme groups are non-planar when oxygen is not bound to it, and the iron atom is out of the plane of porphyrin ring towards that histidine residue. And this is known as the non-planar configuration of deoxygenated hemoglobin. That is considered to be dome-shaped. When oxygen binds to the heme group, it gets that planar configuration where the iron lies in the plane of the porphyrin ring. Okay, and as far as the 5th position, that one is going to be occupied by imidazole, and the 6th position of Fe is occupied by that O2. So you can kind of see the heme group here. This image gives a better explanation or visual to what we talked about in the last slide. We can see the Fe there in the middle, the oxygen bound to it, the center of the porphyrin ring as well. Alright, so the free heme, there's a free heme that is not favored for oxygen transport. Globin in the hemoglobin stabilizes the heme. So furthermore on that O2 complex acts as an effective carrier. And the quaternary structure, which you can kind of see here on the right there, shows a little better explanation. That quaternary structure of hemoglobin is markedly changed on that binding oxygen. So the 3D structure includes alpha-beta dimers that form the tetramers. On oxygenation, the iron moves and brings histidine towards that porphyrin. And in the lungs, all hemoglobin is basically saturated with oxygen, and then when it moves to the tissues, that saturation then drops. So in the picture there on the right, you can see the betas and the alphas. The betas are there at the top and the alphas are there on the bottom within that structure, and we can also see that heme group, that histidine connected there, and then the globin. And this is a picture of that oxygenation curve. So we'll talk a little bit more about this on the next slide. So the oxygenation curve of hemoglobin is essentially sigmoidal by nature, and it exists in the lungs at a high pressure. But when at a low pressure, it passes oxygen to the myoglobin. The O2 need is greatest within the tissues where oxygen is rapidly consumed with the production of CO2. CO2 lowers the pH, causing hemoglobin to release more O2 to that myoglobin. pH sensitivity is known as the Bohr's effect, which we learned in our reading a lot this week. So the H plus increases that acidity, which then decreases that pH. And here's a little kind of image with some explanations that kind of walks you through a little bit more about the Bohr's effect. So we can see the Bohr effect explains how high CO2 helps hemoglobin release oxygen in the tissues that need it. There's an oxygen-loving R form and an oxygen not so loving T form. In the oxygen-loving R form, the His 146 is unprotonated and the T stabilizing salt bridges can't form. And within the oxygen not so loving T form, the His 146 protonated salt bridges stabilize that T state. So I hope this brought a little bit more clarity and can explain a little bit more about the process and kind of how things work and with those pH levels as well. So thanks.